Welcome to Biotanemy.
Plasmid vectors. Cloning vector. A small piece of DNA, into which a foreign DNA fragment can be inserted for cloning purposes. When this piece of DNA consists of a plasmid, then they are known as a plasmid vector. Examples of plasmid vectors are pBR322 and PUC19. pBR322 is a synthetic plasmid vector, which was constructed in the lab of Herbert Boyer at the University of California in 1977. The name PBR stands for plasmid, Bolivar, and Rodriguez respectively, which was named after the inventors of the plasmid, Francisco Bolivar Zapata and Raymond L. Rodriguez. The plasmid is 4,362 base pairs long and molecular weight of 2.83 into 10 raised to 6. pBR322 consists of 1. Origin of replication, 2. Restriction site, and 3. Selectable marker. 1. Origin of replication. The ORI site from PMB1 is used as the origin of replication in pBR322. It has a copy number of 15 to 30. 2. Restriction enzyme sites. 40 different restriction sites are seen in pBR322 plasmid, which are seen in the sites of the tetracycline resistance region and ampicillin resistance region. These sites include HindI3, ECOR1, EcoRV, BAMH1, SAL1, PST1, NDE1. 3. Selectable marker sites. In pBR322, the antibiotic resistance genes are used as selectable markers. 2 selectable marker sites are seen in pBR322. 1 is the ampicillin resistance site. This gene codes for an enzyme that degrades the beta-lactam ring of the ampicillin drug called as β-lactamase. This will allow the plasmid-containing bacteria to grow in the ampicillin-containing medium. If our gene is inserted in any of the restriction site present in this marker region, it will get inactivated and ultimately the plasmid-containing bacterium will not have any resistance to ampicillin, and it will not grow in the ampicillin-containing medium. The other one is the tetracycline resistance site. As seen in the ampicillin resistance gene, the tetracycline gene also works in the same way. The only difference is that, here we use the tetracycline antibiotic rather than ampicillin antibiotics. Insertional inactivation. The selection of recombinant plasmid vector is done using the insertional inactivation method. The steps involved in the insertional inactivation process are: 1. Take the plasmid of interest, say pBR322 plasmid here. 2. Add restriction enzyme which will cut on a particular restriction site or cloning site. For example, PST1, which is seen in the site of ampicillin resistance gene. Also, cleave the gene of interest using the same endonuclease. 3. Add ligases to the mixture containing cleaved plasmid and gene of interest. This will allow the gene of interest to ligate into the cloning site of pBR322. Now, transform the recombinant plasmid into a microorganism which is both sensitive to ampicillin and tetracycline. Note: the principle and procedure for bacterial transformation is already uploaded in this channel. Those who are interested can watch the video using the link given in the description box. And don't forget to subscribe. In step 5. Culture the transformed bacterial cells in 2 different media. 1 with tetracycline. Only the transformed cells will grow in this media, since the original organism is sensitive to both tetracycline and ampicillin. But pBR322 has a resistance gene for both in them. All the cells that grown in tetracycline-containing media is then replica-plated in a media containing ampicillin. The cells that contain recombinant plasmid will not survive in this media, because the resistance gene for ampicillin is lost due to the insertional inactivation process. Hence, the recombinants has been identified, and they will be further subcultured for isolation. The advantages of pBR322 are: 1. Manageable size. 2. Easy to select recombinants, due to the presence of antibiotic resistance genes. 3. Presence of multiple restriction sites. 4. High copy number.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.